A DNA molecule that carries a foreign DNA into a host cell replicates inside a bacterial cell and produces many copies of itself, and the foreign DNA is called cloning vector. Cloning vectors are the next important tools to replication enzymes. Common cloning vectors are plasmids and bacteriophages. Following features are required to facilitate the cloning into a vector. Origin of replication. This is a sequence from where replication starts, and any piece of DNA when linked to this sequence can be made to replicate in the next cells. Selectable markers. In addition to ORI, the vector requires a selectable marker which helps in identifying the transformants and non-transformants. For example, genes encoding resistant to antibiotics such as ampicillin, chloramphenicol, tetracycline are used as selectable markers. Transformation. It is the process by which a DNA piece is introduced in a host bacterium. Cloning sites. These are the sites at which the foreign DNA is combined. Insertional inactivation. Selection of recombinants using antibiotic-resistant genes is a complex process. Therefore, an alternate method is developed. In this, the recombinants and non-recombinants are identified on the basis of their ability to produce color in the presence of a substrate.
Plasmids are good cloning vector as they can carry origin of replication and able to replicate independently within a cell. Vectors must also have a small sequence of base pairs that can be recognized by a restriction enzyme. When the enzyme opens the circular plasmid, foreign DNA can be incorporated. When the plasmid vector and foreign DNA are cut with the same restriction enzyme and mixed together, not all molecules will join to form recombinants. To identify cells that contain plasmids that have incorporated foreign DNA, a second marker gene is needed on the vector. This second marker contains restriction enzyme site within its nucleotide sequence.
Bacteriophage cloning. The recombinant DNA is inserted into the host cell and be purified, then we extract the cells. Step 1. Modification of DNA. The vector is cut by the restriction enzyme. Once the vector is cut, it forms 3 fragments which are 2 lambda fragments and 1 central cluster gene. The target DNA will replace the central cluster gene. Then they are joined together to form recombinant DNA. Step 2. Ligation. Enzyme ligase reacts with ATP to form a ligase AMP complex. It reacts with biphosphate on one side of nicked DNA strand, transferring AMP to the phosphate group. Final stage is detected by free OH forming a new diester bond and releasing AMP, then proceed to the packaging process. Step 3. Packaging. This process is to insert recombinant DNA to bacteriophage. Step 4. Transfection. This is the same as the transformation, but in this we use phage DNA rather than a plasmid.